Assalamualaikum and hello everyone. I am Eka Shafi Zambri from 3rd year student in Biochemistry at University Malaya. Today I'm going to present my topic, which is dendritic cell-based immunotherapy for cancer treatment. For introduction, I will begin with an overview of cancer. Cancer is a disease in which some of the body cells grow uncontrollably and spread throughout the body. Cancer treatment involves a range of approaches aimed to combating the disease. The treatment of cancer depends on where the cancer is, how big it is, and whether it has spread or not, and the patient's general health itself. There are different types of treatment that patient might have, including surgery, radiotherapy, chemotherapy, hormone therapy, ablation therapy, stem cells therapy, nanoparticles, and also immunotherapy. The stage of cancer is determined by the size and the location of the tumor and whether the cancer has spread or not. Cancer stage are indicated by the numbers 0 through 4, which are often written as Roman numerals. Higher number indicates a more advanced cancer stage in the body. The stage of the cancer is used to determine the treatment option and the chances for a cure. For stage 0, also known as a carcinoma in situ, this stage indicates that there are only abnormal cells present with no actual cancer. In short, it means that the cancer has not spread to the nearby tissue. Stage 1, this stage the cancer is small and localized to the one area. It's often referred as an early stage cancer. For stage 2 and 3, at this stage the cancer is larger and has grown into the nearby tissue or lymph nodes but has not spread to other parts of the body. For the stage 4, which is the last stage, this is the most advantage because it indicates that the cancer has spread to other organ of the part of the body. It is also known as the metastatic cancer stage. Now we move to the main part, which is what are dendritic cells. Dendritic cells are professional antigens that presenting cells to inform the fight against the pathogens while enforcing tolerance to safe and harmless avoidable antigens. They capture pathogens and receive signal from pathogens that inform the outcome of the immune response. And now, what is the dendritic cells immunotherapy? Dendritic cells immunotherapy, or DCI, involves using a patient's dendritic cells to create a personalized cancer vaccines. The process typically begins by isolating the dendritic cells to the patient's blood. The cells are then loaded with the tumor-specific antigens which can be obtained from the patient's own tumor or synthesized in the laboratory. Once the dendritic cells are loaded with these antigens, they are reintroduced back into the patients through injections. This process aims to stimulate the immune system to recognize and attack the tumor growth. Now I'm going to show you the example steps involved in the DCI for pancreatic cancer treatment. The first step involves the collection of the dendritic cells. Dendritic cells are typically obtained from the patient's blood or in some cases from their tumor tissue. If derived from blood, the patients undergo a process called leukapheresis. Leukapheresis is a laboratory procedure in which white blood cells are separated from a sample of blood. It's a specific type of apheresis, the more general terms for separating out one constituent of blood and returning the remainder back to the circulations. Now we move to the second part, which is the DC isolation and maturation. Once the white blood cells are collected, monocyte or CD34 precursor cells, which are type of stem cells that can give rise to the dendritic cells, are isolated. The isolated monocytes or CD34 precursor cells are cultured in a controlled environment with the addition of specific growth factor. These growth factors such as IL-4 are crucial for promoting the differentiation or maturation of these cells into the immune system dendritic cells. Now we move to the second steps, which is dendritic cells isolation and maturation. Once the white blood cells are collected, monocyte or CD34 precursor cells, which are types of stem cells that can give rise to the dendritic cells, are isolated. The isolated monocyte or CD34 precursor cells are cultured in a controlled environment with the addition of specific growth factor. The growth factors such as IL-4 are crucial for promoting the differentiation and maturation of these cells into immature dendritic cells. Step 3 is the antigen loadings. The immature dendritic cells are then pulsed with tumor antigens. These antigens can be in form of peptides or proteins that are derived from the tumor cells, tumor lysate, or even whole tumor that have been fused with the dendritic cells. The goal is to prime the dendritic cells to recognize and present the tumor antigens to other immune cells. Now we move to the step 4, which is the activation of the DC. Once loaded with the tumor antigens, the immature DC are matched by the treatment with cytokines such as tumor necrosis factor, alpha, TNFA, and IL-1s. This process will regulate the appreciation of MHC molecules and co-stimulate molecules on the surface of the dendritic cells, which are crucial for the antigens presentation and T cells activation. Now we move to the step 5, which is reintroduced into the patients. Deactivated and antigen-loaded DC are reintroduced into the patient's body typically through injections. These dendritic cells travel to the lymph nodes where they present the tumor antigen to the T cells, initiating for the immune response. This process is designed to activate the patient's immune system specifically against the cancer cells. Now, the last, which is the T cell activation and immune response against the cancer cell. The interaction between the dendritic cells and T cells in the lymph node leads to the activation and expansion of the tumor-specific T cells. Activated dendritic cells migrates from the tumor site to the nearby lymph node and promote for the activation of the cytotoxic T cells, lead to the elimination of the tumor. Now we move to the last part, which is the current research and practice is DCI. There are many benefits of using DCI because it can be customized using various tumor antigens including whole tumor lysate, specific peptides or even RNA and DNA molecules. This flexibility allows the direction of the therapy to different cancer types for individual patients' needs. Other than that, DCI also can act as an educator of the immune system to directly target the tumor antigens. This personalized approach minimizes the damage to the healthy cells while triggering a precise immune response against the tumor cancer cells. Unfortunately, the success of the DCI for cancer treatments are still being evaluated, and the clinical benefits are considered suboptimal and unsatisfactory. There are many ongoing research and clinical development are focused on improving the immunotherapy efficiency of the DC-based treatments, particularly through the development of the combinatorial approach, patients' new vaccines, and a better understanding of the DC function of the cancer treatments. That's all from me. Thank you.